No, I thought today was your day to get the milk.
No, yesterday was my
Hey,
day.
guys.
Hey Zach, I really like your red shirt. It matches your father's hat.
What are you talking about? That's red. This is blue.
No, that's red and that's also red. Right, honey?
Yeah.
No, this is red and this is blue.
Oh, we're colorblind. I guess we just forgot to tell you.
Oh, that's weird. Why didn't you tell me before?
I guess it never came up. I mean, we were both born with color blindness, so we see blue as red.
Oh.
Yeah.
That's weird.
Oh, hey, since your father forgot to pick up the milk, can you go run out and get some milk for us?
Yeah, sure.
Thanks. Hello, class. How are we doing today?
Doing well.
Glad to hear it. So today we're going to learn about sex-linked traits. So, a sex-linked trait is a trait only found on the X chromosome. Therefore, females will have 2 copies of the sex-linked trait, while males will only have to have 1 copy of the gene. Since male cells are XY and female cells are XX. Because males only have 1 X chromosome, they have a greater chance of receiving the sex-linked trait. This is because for a recessive trait, females can have 1 copy of the trait and still not show the disorder, making them heterozygous and a carrier. Meanwhile, if males have 1 copy of the allele for the gene, they will show it since they only have 1 X chromosome. An example of sex-linked traits include hemophilia and colorblindness. Now, we will do a Punnett square showing colorblindness. As you can see in this first Punnett square, we're doing a cross between a female who's a carrier for colorblindness, so she only carries 1 allele for colorblindness, and a male who is colorblind. If they were to have children, they would have 1 female who is colorblind, another female who's a carrier for colorblindness, a male who is colorblind, and a male who is not colorblind. Now if we look at the second Punnett square, we are going to do a cross between a female with colorblindness, so she carries 2 alleles for colorblindness, and a male with colorblindness.
Hey, that's just like my parents.
I didn't ask, so please do not disrupt the class again. If these 2 were to reproduce, however, all of the children, regardless if they were male or female, would all be colorblind.
Wait, that means...
Honey, did you pick up Zach from school?
Oh, today was your day to pick him up.
No, yesterday was my day. Today was your day.
Guys, you never picked me up from school. I had to walk all the way here.
Oh my God, blame your father for that one. Oh my God, I can't believe you forgot that milk. How was school?
It was fine, but in bio today, we were learning about sex-linked traits and colorblindness and all that.
We're colorblind.
And with the Punnett square, they showed how if both parents are colorblind, all the children have to be colorblind. And it was the weirdest thing, because both of you are colorblind, and I'm not colorblind, so...
Oh, you know, well, your teacher's dumb. She doesn't know what she's talking about. I mean, what, she went to Harvard? That's not even a good school.
No,
I mean,
it's not, yeah.
I don't think you should listen to her.
No,
She's just trying to mess with your head,
Yeah,
you know?
she is, I know.
Honey,
Yeah.
can I talk to you for a minute?
Sure. Okay. Alright. Bye.
Honey, what should we do about this? He's getting so close to our secret. He cannot know he's adopted.
No, we can't tell him.
I think it's best if we don't tell him, because it's for his own good, right?
Yeah.
We're just looking after him like a true parent.
Yeah, there's no way we're gonna tell him.
No way.
Okay.
Okay, just
Agreed.
keep it on the down low and we're
Yeah.
all good.
Good.
Okay. Hello class, how are we doing today? I don't care. So, today we're going to learn about gel electrophoresis. And gel electrophoresis is a technique for separating protein molecules of different lengths. The small molecules of DNA will go the furthest and the fastest, while the larger strands won't go as far. The way to do this is by placing the DNA through a block of gel like Argos, and placing the DNA on the negative side of the gel, and run an electric current through it, so that the DNA will travel to the positive side. Yes, Zach?
What would you use this for exactly?
I mean, I guess like paternity tests, things like that. It's pretty useful in the scientific field.
I'll use that on my parents to see if they're actually my parents then.
Are you kidding me, Zach? You've interrupted my class one too many times. Get out of here.
Oh.
Go to the principal.
Honey, did you remember to do the dishes? Today was your day.
Yesterday was my day to do the dishes. Today was your day to do the
Hey
dishes.
guys, hey.
Hey.
So,
Zach, how was school today?
it was good. It was good. I just need a little hair sample from you guys. You know, for a science project.
A science project?
Yeah.
What kind of project is that?
It's for bio. Yeah, you
Oh,
know,
that
it's
teacher.
no big, that teacher. You know, the Harvard one.
Yeah.
The dumb one, yeah.
Yeah,
Okay.
yeah.
I guess that'll
So
be
just,
okay.
just whenever you can.
Okay.
Here you
Oh,
go.
okay, that works. Okay, thank you guys. See ya.
Alright.
I'm going to my friend's house. Bye.
Hi, how are you?
Hi.
So, by the hair strands that you gave me, I originally thought that I
Okay.
would have to use PCR
Okay.
because I didn't think that there was enough genes on the hair strands.
Okay.
Turns out there were, so I didn't have to use that anyway.
What's PCR?
PCR amplifies DNA,
Oh.
and you could use it in like crime scenes and
Mhm.
stuff like that. But anyway, I performed a gel electrophoresis test,
Mhm.
which is a form of lip analysis,
Yes.
and turns out, unfortunately, that your bands don't match up with your parents' bands.
Oh, what does that mean?
Means that you're adopted.
Oh yeah.
The sun is an important part to an ecosystem. It provides energy to plants during photosynthesis. In the light reactions of photosynthesis, the sun's energy is used to break down a water molecule in the process of hydrolysis. From this, hydrogen ions are made and also oxygen molecules that are then released into the atmosphere. Each photoexcited electron passes from one primary electronic acceptor into photosystem 2 to photosystem 1 via an electron transport chain. As electrons move down the chain, their exergonic fall to a lower energy level is harnessed by the thylakoid membrane to produce ATP by chemiosmosis. The production of ATP in the chloroplast is called photophosphorylation because the energy harnessed in the process originally came from light. This process of ATP production is called non-cyclic photophosphorylation. The ATP generated in this process will provide the energy for the synthesis of glucose during the Calvin cycle, or the light-independent reactions. In the Calvin cycle, there are 3 stages: fixation, reduction, and regeneration. In fixation, the first stage of the Calvin cycle, light-independent reactions are initiated. CO2 is fixed from an inorganic to an organic molecule. In the second stage, ATP and NADPH are used to reduce 3-PGA into G3P. Then, ATP and NADPH are converted to ADP and NADP+, respectively. In the last stage of the Calvin cycle, Ruby P is regenerated, which enables the system to prepare for more CO2 to be fixed. With 6 turns of the Calvin cycle, a glucose molecule will be made, and this will provide the starting point for cellular respiration.
In food chains, the predator only gains 10% of the prey's energy. The amount of energy at each trophic level is transferred to the next level. The rest is lost through metabolic processes. We see an example of this when this bunny rabbit consumes the grass, as well as when the hawk consumes the rabbit. Each level of the food pyramid only gains 10% of each prey's energy, and because of this, the lower on the food pyramid a certain prey is, the more the predator has to eat, while the higher on the food pyramid a certain prey is, the less the predator has to eat.
Honey, did you remember to feed the dog today?
You have a dog?
Oh my god, you forgot to feed the dog?
I didn't know that we
Well,
had a dog.
how can you forget
How
about
is
the
that
dog?
my fault?
Now the
I
dog's
told
dead.
you
Oh my
to
gosh,
feed the
you
dog.
killed our dog. Hi, Zach. What's the matter?
I found out something today.
About the dead dog? Yeah, I know, blame your father for that one.
We have a dog?
See? Oh my god, guys! Well, not anymore because it's dead, but anyway, what's the matter?
Well, so I went to the doctor's office today, and, well, she ran a gel electrophoresis test with those hair samples that I got, you know, for the science experiment. And, well, the results said that I'm adopted.
Well, Zach, so, about that. We've been meaning to tell you, we love you, As if. Don't cry. Don't cry. You're gonna make me cry. We love you as if you're our child. Just understand that we had your best intentions at heart, okay? We love you. And just because you're adopted doesn't make us love you any less. Right, honey?
Yeah.
We love you.
We love you.
We love you.
I love you guys.
So that's today's lesson. And I'll see you guys tomorrow. Honey, did you remember to feed the dog today?
We have a dog.
Hello, class. How are we doing today? I don't care. So, today we're gonna
Hey,
Zach.
No, Zach.
Forgot.
Okay.
That's really
You're
angry.
angry.
Sorry.